Hello, in this video we'll be discussing which corn product is best for fermentation and a walkthrough of our experimental procedure. The goal of this experiment is to figure out which corn product is the best for fermentation, specifically between stem extract and kernel extract, and hopefully applying our research will further our understanding on the efficiency of using corn for biofuels. In order for fermentation to occur, it requires glucose to begin glycolysis. Corn is rich in starch, which contains alpha-glucose monomers, and cellulose, which contains beta-glucose monomers. Therefore, we can assume that stem extract and kernel extract will ferment to some extent. Moreover, we plan to use corn syrup, a substrate where known fermentation will occur, since it contains sugars like dextrose and maltose. We'll be using the corn syrup as a positive control group. To begin the experiment, we first label 5 dirham vials to ensure accuracy while testing. Next, we prepare 5 milliliters of stem extract, kernel extract, corn syrup, and water into each vial, and pre-incubate them for 10 minutes at 30 degrees Celsius. Then, we swirl the flask containing the yeast to resuspend it, and add 2 milliliters to each tube. After vortexing each vial for 10 seconds, we fill 5 different tubes with each mixture using a pipette and put them in upside down in the vial. Now we return the apparatus to the water bath and measure the amount of CO2 in the tube every 5 minutes for a total of 45 minutes. The results from our experiment showed that the corn syrup positive control group produced 4.2 milliliters of CO2. The kernel extract produced 3.3 milliliters, the stem extract produced 1.1 milliliter, and finally the 2 negative control groups produced no CO2. The rate of production for the CO2 for the corn syrup was quick, producing 2.7 milliliters after 5 minutes and an additional 1.7 after 10. The kernel extract produced 1.8 milliliters of CO2 after 5 minutes and then produced a steady rate of 0.7 to 0.8 milliliters of CO2 every 5 minutes for a total of 10 before plateauing at 3.2 milliliters. The stem extract was much slower, only producing 0.2 to 0.1 milliliters of CO2 every 5 minutes for a total of 30 minutes before eventually slowing down at 1.1 milliliters. To summarize, the corn syrup had a linear rate of production of 0.3, the kernel extract had a linear rate of production of 0.14, and the stem extract had a linear rate of production of 0.04. The 2 negative control groups had a linear rate of production of 0. We can immediately tell from this experiment that corn syrup is the best at fermentation. This is an expected result, however, thanks to a study done by Steinkraus and Robinson in 1967. Between the 2 experimental groups, the kernel extract is clearly superior in fermentation, producing 3 times as much CO2. This is most likely because the kernel extract contains more starch and cellulose, both of which contain glucose monomers. Starch and cellulose aid the fermentation process, ultimately producing more CO2. Finally, we see that 2 negative control groups produced 0 CO2. This is an expected result, since they shouldn't ferment without yeast. To conclude, the kernel extract produced more CO2 at a faster rate than the stem extract. This is due to kernel extract having more starch and cellulose than the stem extract. If we were to apply our findings to the study of biofuels, we can assume that kernel extract would be an ideal corn product for fermentation. Thank you for watching this video.